Today I'm going to talk about the use of aborted fetal tissue and cells and DNA in vaccines. And the title of this talk is, you can't be, if you're pro-life, you can't be pro-vaccine. So Johnny, if you would hit the next slide, please. So this is a slide that shows the vaccines that are manufactured with human cell line and alternative cell line products in the USA. And this is actually this came from Sound Choice Pharmaceutical Institute, and you can find this information at soundchoice.org. And if it's difficult to see, the numbers, the vaccines right now that are on the United States childhood schedule, which contain aborted fetal tissue DNA, cells and DNA, are the chickenpox vaccine, and that contains WI38, which is from a female, and MRC5, which is from a male, hepatitis A and B, the combo shot, the MMR vaccine, the MMR plus chickenpox, which is called Proquad, the Pentacell vaccine. A lot of people don't realize Pentacell also has MRC5 cells in it. So those aborted fetal tissue, aborted fetal DNA in the final product that is injected into our children. And most people know that the MMR vaccine and the chickenpox vaccine have, many people do anyway, a lot of people don't know, but many people who are sort of in this movement understand that it's in the measles vaccine and the varicella vaccine, but Pentacell is given at 2 months, 4 months, and 6 months of age, so it's actually being injected much earlier than it would be with just the MMR and the varicella vaccine. And Pentacell covers DTaP and inactivated polio and Haemophilus B. So, and then the shingles vaccine, which is not on the childhood schedule, but on the adult schedule. Okay, Johnny, please. And now this is the childhood schedule in the United States. This is the CDC schedule for children from birth to 2 years of age. And it shows MMR and varicella vaccines are given at 12 to 15 months of age. And you can see that, let's see, MMR, this is measles, mumps, rubella vaccine. And then you can see right here, this is the first dose, which is at 12 to 15 months. And they also give varicella vaccine at 12 to 15 months. Now these vaccines are also often given, frequently given at the exact same time, even though the MMR vaccine manufacturer's insert says that it should never be administered along with other live virus vaccines simultaneously, but it's done all the time, along with not only that, but hepatitis A vaccine, which is also a live virus vaccine. Next slide, please. Okay, so this is, you know, we think about what's the incentive behind all of this. This is a graph showing, or a table, showing the top 5 vaccine products worldwide. It's projecting what the sales would be by 2020. And you see Prevnar 13 up here is the one that's projected by 2020 to be the highest grossing vaccine in the United States, or in the world. Pentacell is down here. That's Gardasil is number 2, by the way. Pentacell down here by Sanofi Pasteur is projected to, with sales at more than $1.6 billion per year by 2020. And Pediatrics right here, this is the number 5. That is, that's by GlaxoSmithKline. That one's also $1.5 billion projected by 2020. That's another 5-valent vaccine. Next slide, please. Okay, so these are the top 10 companies, total and worldwide vaccine sales, 2014 and then projected in 2020. You see the one up here, it's hard to see, I know, but it's Merck and Company. And their projected sales by 2020 are projected to be about $7.5 billion per year. And Merck is the one that makes the most of the vaccines that are on, the biggest number of the vaccines that are on the childhood schedule in the United States. They make the MMR vaccine and varicella vaccine, and they make several others. So they're the ones that are making the biggest bucks. Now down here you'll see the total industry in 2014 was almost $27 billion, and then by 2020 it's projected to be almost $35 billion per year. Next slide, please. So when we talk about the abortions, these abortions were used in the manufacture of vaccines, and they're still being used in the manufacturing of vaccines. This tells us about how the abortions were done for the original vaccines. And this comes from Dr. Peter McCullough's book, The Fetus as Transplant Donor. the scientific, social, and ethical perspectives, reports on the methods used in harvesting fetal tissue in Sweden. Now, Sweden was where the abortions were done, and then the tissue and cells were utilized by United States vaccine manufacturers. They would puncture the sac of a pregnant woman at 14 to 16 weeks, put a clamp on the head of the baby, pull the head down into the neck of the womb, drill a hole into the baby's head, and attach a suction machine to remove the brain cells. At 16 to 21 weeks, they would do prostaglandin abortions, where a chemical is injected into the womb, causing the woman to go into a mini labor and pass the baby. 50% of the time, the baby would be born alive, but that didn't stop them. They would simply open up the abdomen of the baby with no anesthesia and take out the liver and kidneys, etc. Make no mistake, these abortions were done this way to ensure intact organs and tissues for research. And you can find this information down here. The DVD is available at cogforlife.org/DVD/. Next slide, please. Okay, when we talk about the use of aborted fetal tissue in vaccines, one thing that comes up is that the Catholic Church has approved this because the benefit of vaccines using these aborted fetal tissues outweighs what? The risk to our souls? The benefit makes it worthwhile is basically what they say. I want to say something about the abortions before moving on. Paul Offit says there were only 2 abortions involved in the development of the vaccines using fetal tissue, and they happened in the 1960s. Dr. Offit says that's all they are using. Technically, there were 2 abortions that were used in developing MRC5 and WI38. Those are the cell lines. Those are the names of the cell lines. MRC5 is from a male aborted baby. WI38 is from a female aborted baby. So we've got both male and female DNA. Just hold on to that for a thought. But here's the thing. Before they developed those cell lines, they had to find babies that were infected, for example, with rubella. This was in the 1960s. There was a rubella outbreak. They wanted to develop a vaccine, so they scared pregnant women by telling them that their babies had been exposed and they would be born with horrible birth defects. So they convinced 27 women to abort their babies before they found one baby who was infected with rubella. That's why the strain that was used in the development of the rubella vaccine is called RA273. R is for rubella, A is for abortion, 27 is the number of babies, and 3 is the number of tissue samples taken. Altogether, there were more than 80 aborted babies involved in the development of the rubella vaccine alone. So when Paul Offit tells you there were only 2, he's lying, because he's a lying liar that lies. Another issue here is that fetal cell lines are problematic because they are highly tumorigenic, meaning they cause cancer. The more times they replicate, the older those fetal cells become, and the more times they replicate, the more tumorigenic they become. So that's a real problem when we need to talk to Paul Offit about, since the cell lines currently being used in vaccines injected into our children were developed in the 1960s. So when he's telling us, oh, don't worry about it, it was so long ago, that's a huge problem, because the older those cell lines are, the more likely they are to cause cancer in our children who are being injected with them. Next slide, Johnny. Okay, so let's talk about the benefits of measles vaccination. What? The benefits of measles vaccination. Measles is a mild ailment. This was a report on measles outbreaks in the UK from 1959, before we had the vaccine to inject fear into measles, what was a mild childhood illness. So this is a report from the British Medical Journal, published in 1959, and they went out and they talked to doctors who had been treating and seeing patients in this big outbreak of measles in the UK, in England and Wales. So in this particular report, Dr. Fry says, measles is a mild ailment with very few serious complications at any age, and all children have made complete recoveries. As a result of this reasoning, no special attempts have been made at prevention, even in young infants in whom the disease has not been found to be especially serious. well and truly over in a week from the prodromal phase, which means before you get the rash, and the disappearance of the rash, and many mothers have remarked how much good the attack has done on their children, as they seem so much better off after the measles. Mothers were reporting that their children had these big developmental gains after they got over the measles. And in this report, he talks about how babies as young as 7 months of age had no complications. So it's all about fear. Once we had the vaccine, that injected the fear into the measles. So when we bring up the fact that in developed nations like the UK and the United States, the death rate from measles had decreased by 98 to 99% before the vaccine was ever introduced, we are often accused of being calloused and not caring about babies in Africa, because everyone knows measles is deadly in Africa, right? Well, let's take a look at what measles was really like in Africa way back in 1979. Next slide, Johnny. Measles is deadly in Africa, right? No. This slide shows, this is from Zambezia, which is in Africa, 1979. This is from the medical journal, a natural history of measles, from the Department of Pediatrics and Child Health, the University of Rhodesia. And the author says, "You may wonder why I have taken such a mundane disease as measles for the subject of this lecture. Most people regard it as a mild illness, often no worse than flu. Parents may welcome it as something inevitable, while for many children it means nothing more than a forced holiday. It is a disease of which most of us have personal experience, and therefore I hope is of interest. Many of us possibly retain vivid memories of our own attack and the way in which it was treated. My main recollection is of the darkened room in which I was nursed, and later of my being forced to wear a cap with a green lining specially bought for the occasion to protect my eyes from sunlight." And you can find this article at tinyurl.com/measles Zambezia 1979. Next slide. This is from the same article, and it shows the clinical course of natural measles infection. And what it says, this down here, I've written up here, I believe, immunity from natural measles is probably lifelong. No, actually, this is from another part, but what this says, immunity from natural measles is probably lifelong, but lasts at least 64 years. That's important because with the vaccine, there are two issues with vaccine failure. There's primary vaccine failure, when the vaccine does not produce any immunity in the first place, and then there's secondary vaccine failure, in which any immunity that is conferred initially wears off within 5 to 20 years. And that's a big problem, because if we're vaccinating our children at 12 to 15 months of age, and again at 4 to 6 years of age, and the vaccine wears off in 5 to 20 years, then we are leaving adults more vulnerable to infection at a later age, including women during their childbearing years. So what we have done as a result of the vaccine is we've taken what was a mild childhood disease, and we have shifted the age of vulnerability to an age when the consequences are more serious. And we have the vaccine to thank for that. Now over here in this, it says the clinical course of natural measles infection. Number 1, the measles rash appears 12 to 14 days after contact. Its greatest infectivity is during the late prodrome, 3 to 4 days before the rash appears. The disease is contagious, probably spread by droplets, and not miasmic in origin, which means it's not smelly. The protection from an attack is lifelong. The last epidemic of measles in the Faroes had been in 1781, and I can't read that name, but found that the only inhabitants immune were those over 64 years old who had suffered from the disease as children during the earlier epidemic. So we know that getting measles naturally confers long time, probably lifelong immunity. Yes?
It looks like measles is cyclic precisely because of that immunity.
Yes.
So you get a generation, and then it's another generation goes before you get another.
Yes, there are natural cycles of illness with measles, just as there are with whooping cough. Very good point.
Could you repeat that question?
Gentleman says it looks like the disease is cycling with outbreaks of measles dependent on the immunity of those who suffered an earlier attack. So, and that's the natural course of illness, when we have a natural immunity from gaining the infection and getting over it. There's much greater immunity, so we have fewer attacks. And when we have those, they boost the immunity of those who were around. Same thing with chickens as well. Next slide. This is from the Lancet. Everybody know that rag? This is, they talk about, don't show me any of those studies from those lower tier journals. You can't get much more highly respected top tier than the Lancet, right? Yes. So the Lancet in 1985 reported measles, Johnny, go back one. There we go. Measles virus infection without rash in childhood is related to disease in later life. Now what they found was, this study was looking at two groups of adults. And one group consisted of adults with history of natural measles infection in childhood. The other group were adults who either had no history or evidence of natural measles, or who had evidence of measles by blood test for specific IgG measles antibody, but no history of clinical measles. So they may have been exposed, but they didn't develop the rash. And that's what we see a lot of times in our children who are vaccine injured, is that they have evidence of chronic measles infection from the vaccine, but they never produce the rash. Okay? So what this is looking at, what they were looking at was to see, they were looking at the incidence of disease in adults to see if there was a difference between the two groups. And you can find this study at tinyurl.com, Measles Lancet 1985. Next slide. So let's have just a tiny little lesson here about statistics and how to read these results. In research, we are looking for statistically significant results. P, the little p, means probability. This is called the p-value. It's a measure of the strength of your results, the power of your study. When you're talking about probability, 0.05 is what is generally accepted as being statistically significant. That means that if you're looking at 100, p is less than 0.05, that means there's a 95% probability that what you're seeing is real and not by chance. Now, p-value of less than 0.01 means there is a 99% probability that what you're seeing is real. When you get to p-value of less than 0.001, that means there is a 1 in 1,000 probability that what you're seeing is by chance, and that is really strong statistical evidence. Okay? A little lesson there. So can measles be good for you? This is from the same study, the Lancet study. And this is the chart or the table that reports their results. They found significant results. They looked at different disease states in adults. What they found, they looked at immunoreactive diseases, which is autoimmunity, sebaceous skin diseases, which is eczema and psoriasis and those kinds of things, tumors other than skin and cervical cancer, and degenerative diseases of bone and cartilage. And what they found was that there was a highly statistical significant difference between the two groups based on whether or not they had natural measles as children. And they found that those who did not have measles were far more likely, at a p-value of less than or equal to 0.008, which is 8 out of 1,000 chance that you are seeing something that's not real, right? For immunoreactive diseases, autoimmune disease, sebaceous skin diseases, p-value of 0.001. Tumors, so cancers, p-value of 0.001. Degenerative diseases of bone and cartilage, p-value of 0.005. That means that these people, so down here, what does this mean? Those who got measles naturally as children had much fewer incidences of chronic debilitating diseases as adults. They were protected against autoimmune disease, cancer, degenerative diseases of bone and cartilage, and serious skin diseases, all those things we now have epidemics of and which were much less common prior to mass vaccination with MMR. And again, you can find this study at tinyurl.com, Measles Lancet 1985. Next slide, please. Okay, so let's just recap what do we know so far. We know that many babies have been aborted and continue to be aborted for the sake of developing vaccines. We know that vaccine manufacturers are making billions of dollars from the sale of vaccines made from babies who were killed. We know that measles was considered a mild, uneventful childhood illness in the decade prior to the licensure of the first measles vaccine, even in Africa. We know that getting measles as a child is protective in adulthood against cancer, autoimmune disease, sebaceous skin diseases, and degenerative diseases of skin and bone. So let's talk now about what injecting the DNA of aborted babies does to children receiving those vaccines. I want to say something before we move on. Up here I say, that they continue to be aborted for the sake of developing vaccines. In 2015, there was a new fetal cell line developed, and it's called Walvax-2, WALVAX-2, and you can look it up online. It was done, the abortions were done in China. They are developing, there's a race to develop a new fetal cell line to replace the MRC5 cell line because they know that it is becoming increasingly tumorigenic and needs to be replaced. So Walvax-2 was developed in China, and they aborted 9 babies. They used a water bag abortion, which is illegal in the United States. They used a water bag abortion where they inject saline into the womb to develop the baby in the sac. They were looking at babies up to 20 weeks, 22 weeks of age, the fetal development. They developed the babies whole in the sac so that they could be dissected and their organs left intact for research. This is a new cell line. This is ongoing. This is not about 2 aborted babies in the 1960s. This is an ongoing thing. Okay, next slide, Johnny. Okay, so this is some information that I've added since the previous version of this PowerPoint. I just did a Google search. I wanted to see, because people said, well, how do you know the government knows about this? So I did a search, and I found PowerPoint presentation, issues associated with residual cell substrate DNA from the FDA, and this is from November 16th, 2005, so 12 years ago. Almost 12 years ago, and then there's an updated PowerPoint from the FDA, and so I looked at these two. All right, so this is from the 2005 draft. This is you can see it there, it says draft, from Keith Peden, the Division of Viral Products, Office of Vaccine Research and Review, from the FDA. And the title of this is Vaccines and Related Biological Products Advisory Committee, from November 16th, 2005. Title is Issues Associated with Residual Cell Substrate DNA. That's the cover. So let's go to the next one. Now this is the cover slide from the updated PowerPoint in 2008 from the same guy, Keith Peden, from the FDA. New cells for new vaccines, September 30th, 2008. And again, issues associated with residual cell substrate DNA. An update. Next slide, Johnny. All right, this presentation this is the 2008 presentation. So there had been concerns about residual DNA in vaccines for 40 years at that time. Because they're saying, the aim of our studies to answer a 40-year-old question. Can residual DNA from the production cell substrate pose a risk to vaccine recipients? They've been concerned about this for, at that time, in 2008, 40 years. So now we're nearing the 50-year mark. Wouldn't it have been nice if these questions had been answered before injecting the entire population of the United States? Next slide, Johnny. All right, and up here, you can see I've kind of pointed to that, and I know you can't see it here, but you can, if you find the slides, you can go find these things online for yourself, and the address is here. All right, so the World Health Organization and the FDA set limits for the amount of residual DNA from cell lines because of concerns about DNA causing cancer. Cell lines, cell substrates, and WHO recommendations for DNA limits for parenterally administered vaccines. Parenterally means injected, right? Cell lines, less than 10 nanograms per dose. That's the level set by the FDA. Next slide, Johnny. All right, so issues remaining to be addressed, and this is, again, from the 2008 study. Issues remaining to address. Whether cell-strate DNA is oncogenic, oncogenic means causing cancer, in our experimental system. Guess who the subjects are in this experimental system? Our kids. And the need for a positive control. They want to eliminate that positive control, don't they? Let's get rid of the unvaccinated kids so we don't have that. Contribution of the host immune system to DNA-induced tumor formation. So what does that mean? The host immune system. That means individual differences, and if some people might be more vulnerable than others. biological activity of chromatin, routes of inoculation, oral, nasal, and then frequency of DNA-induced initiation event. Now down here, they want to know whether heritable epigenetic effects can induce oncogenic events in vaccine recipients and whether these could pose a safety concern. Again, wouldn't it seem prudent to figure out these concerns before injecting fetal DNA into every child in the United States? For what? To prevent measles and chicken pox? And it's not just cancer. Johnny, next slide. All right, so this, there's no way to read this, but you can go to tinyurl.com, COG4Life, the number 4, Vax Autism. This is a study from Sound Choice Pharmaceutical and Dr. Teresa Deischer. The researchers looked at MMR, varicella, and hepatitis A vaccines, and they wanted to see how much fetal DNA is in the vaccines. The FDA limits fetal DNA in vaccines because it's dangerous, and we've seen that they've had concerns about this for almost 50 years. The researchers found that the vaccines contain much higher amounts of fetal DNA than the FDA allows, and the type, which is fragmented DNA, is much more easily inserted into the nucleus of the cell of the vaccine recipient. They also found hotspots for recombinant DNA, which is that DNA inserts into it and it recombines with the host DNA, recombinant DNA on several genes that are associated with autism. Next slide, Don.
Can I ask a question here? I'm of the generation, I think the few little vaccines I have, they were done with monkey tissue, okay?
The MMR was not done. You're thinking of the polio vaccine. Yes.
Polio,
Yeah.
smallpox.
And there are a lot of other vaccines that are not made with fetal DNA frequently contain the DNA of animals. So yes, that's another issue, and you're right. Yes. Okay. So this is from the summary of that the study that we were just looking at, the homologous recombinant DNA study from Sound Choice Pharmaceuticals. And I'm just going to summarize this down here. The amount of residual fetal DNA in vaccines was tested at 14 to 27 times the FDA limit for single-stranded DNA and greater than 3 times the limit for double-stranded DNA. So there is way more fetal DNA in the vaccines than the FDA has set the limit for. Up to 27 times the amount in the vaccines that are being injected into our children. Next, Johnny. So this is another study from Sound Choice Pharmaceutical, and this is tinyurl.com/cog4life cancer, Vax cancer. This study furthered the investigation into the uptake of fragmented fetal DNA in vaccines. They found that both damaged and healthy cells spontaneously incorporated fetal DNA fragments into the nucleus very quickly. And this alters the DNA via a process known as insertional mutagenesis. Insertional mutagenesis was studied with stem cells for children. And they looked at, there was a study done several years ago with looking at insertional mutagenesis from stem cells. So these children who had very serious immune system deficiencies they had stem cell transplants. And within 4 years of being injected with the stem cells, a majority of those children developed leukemia. And it was found to be due to insertional mutagenesis. And insertional mutagenesis is exactly what's happening with the DNA from vaccines. It's doing the same thing. It's incorporating into the cells of our children, and it's causing increases particularly in lymphoma and leukemia in children. Next slide, Johnny. All right, so this slide is about the legislation and what's happening and why our legislators are pushing for mandates for vaccines. This lady here is Senator Patricia Miller. I live in Indiana. She's one of our senators there, or was. She retired last year. She voted for HB 1337, which makes it a level 5 felony to acquire, receive, sell, or transfer fetal cells in Indiana. In 2016, Senator Patricia Miller introduced SB 162, which sought to mandate all adults in Indiana to receive MMR and varicella vaccines, if they work in healthcare or if their job takes them into a hospital or clinic setting. Dear Senator Miller, do you realize if SB 162 passed, you would be making all healthcare workers in Indiana felons? What make a legislator go against his or her pro-life stance on this issue? Next slide. Women in government. Everyone needs to know about women in government. Women in government is basically a private club of lady legislators who are heavily courted by the pharmaceutical lobby. They wine them and dine them. They hold conferences in five-star hotels, multiple conferences throughout the United States every year. They pay their airfare, ground transportation, hotels, food, drinks, everything, and they get them in a room and they present their information so that they have the information the pharmaceutical companies want them to have to bring up the legislation to promote the pharmaceutical products. Patricia Miller is a Republican, and she was the chair of the Senate Public Health Committee, and Cindy Kirchhofer was the chair of the Public Health Committee in the House. These are women in government. Each state has state directors, and these are two of the state directors for the state of Indiana. Next slide. These are screenshots from the Women in Government website, and this is the 2015 sponsors list. And they include Merck, GlaxoSmithKline, Novartis, Pharma, etc. And there's Eli Lilly in there, Eli Lilly, Bristol Myers Squibb. So I think AstraZeneca is on there. But anyway, they used to have on their website, Women in Government used to have a statement.
The American Chemistry Council.
Yeah, so you get the idea, right? Lots of sponsors, and these sponsors are basically paying our legislators to listen to their propaganda. And they used to have a blurb on the front page of their website that said about us, and in that they just said our mission is to promote legislation that is favorable to our sponsors. These are our elected officials. And they are promoting legislation that is favorable to their sponsors, not to us. Next slide, Johnny. All right, so I already told you there, Senator Miller was the chair of the Senate Public Health Committee, and Representative Cindy Kirchhofer, was the chair of the House Public Health Committee. In 2016, Patricia Miller had control over the Senate Public Health Committee, and Cindy Kirchhofer maintains control over the House Public Health Committee. These two lady legislators have control of what bills get heard and what doesn't get heard. If a bill is introduced that is not favorable to their pharma sponsors, they have the power to prevent it from ever being heard. How is that legal? Pharma owns our state house. Next slide. This is also a screenshot from the Women in Government website. Dr. Gregory Poland is a vaccine scientist. He's the editor-in-chief of the journal Vaccine. He's also a Merck representative. He works for several vaccine manufacturers. He founded the Vaccine Research Lab at the Mayo Clinic. He is a paid consultant for multiple vaccine manufacturers, including Merck, which makes the MMR and varicella vaccines. Dr. Poland was a guest speaker at the January 2016 Women in Government State Directors Conference, where he delivered this presentation on how to increase vaccine uptake among adults in the United States. And here's where you can find this presentation in its entirety, tinyurl.com/millions4 mandates. Next slide, Johnny. So this is slide 33 of 37 in Dr. Poland's PowerPoint presentation. He offered a $1 million bribe to state directors of Women in Government to introduce and pass legislation to mandate vaccines in their states. We all need to be looking at the voting records of our legislators and at the campaign contributions, as well as their association with women in government. Call them out for their hypocrisy and expose them. Register with NVIC, advocacy.org to track legislation and fight for the rights in your state. Now, what this says, this is directly, and it's his typo here, just as it was left. If a private donor could be identified, we would provide each of you with a $1 million grant, in parentheses, his parentheses, if you develop legislation and policies that materially improve the health of your states and communities. Could you do it? This is about vaccines. The entire presentation was about vaccines. So that's a bribe. This presentation was delivered in January of 2016, and I think it was within 6 weeks of this, there were multiple bills, 12 bills, new bills in multiple states introduced to mandate vaccines for adults, and they were introduced by state directors of women in government. So we all need to be watching out for this. Call them out, expose them, make flyers, and put it on every telephone pole and every billboard in their constituency, in their district, and expose them for the liars that they are.